Okay, one more minute. Okay, time's up. So just submit whatever you have, and we will start with today's class.
Okay, so yeah, today's class is going to be interesting. So today we're going to talk about the so-called reproducibility crisis in science, in general, but specifically in the biomedical sciences. Okay, so first, how do people define the reproducibility crisis? So it is an ongoing methodological crisis in which it has been found that the results of many scientific studies are impossible or difficult to reproduce. Okay, so this is the definition. This is very clear, right? Now, when we are assessing reproducibility, we're actually talking about three different concepts. Reproducibility, which actually means being able to find the same results using the same data and the same method. So if you apply the same methods, if you ask someone else to apply the same method you applied on your own data, they should find the same result. That's the first one. The second one is robustness, which actually means if you have a set of data, for example, in our field, we have sequence data, and I run a variant calling program and say, oh, this is the set of mutations in my data, and I give my data to someone else that find mutations, and they run a different program and a different set of methods with the same objective of finding mutations. If my methodology was robust, they should find something similar. The methodologies are different, but the objectives are the same, and the data is the same. Therefore, that should replicate. That's robustness. And then finally, replicability is more broad. If you claim, for example, there are these mutations associated with this type of cancer, and I did my study, and here's my data, and here's my method. Someone else can go and say, I'm going to study the same type of cancer, different people, different methods, and I should find the same, because that's your finding. Right? So these are really the three different levels, let's say, of replicability that we will be discussing. Okay, so the question really is there a reproducibility crisis in science? As we saw yesterday with the funnel plots and lots of people studying this, it's actually difficult to assess if this is the case. It's complicated because sometimes studies are not exactly the same. They have slight differences in objectives, for example. So people say, well, they're not comparable. I do not know exactly if this is reproducing the other one. So it's not that easy to know. But what Nature did, this is a poll for scientists. It was done in 2016. And they asked more than 1,500 people working in different areas of science if they thought there was a reproducibility crisis. So more than half said yes, a significant crisis. And another significant chunk of these people said yes, a slight crisis. Right? So most people, most scientists in working in research actually think there is a problem with reproducibility of results of papers in science. Now, this varies by field, of course. If you can see here, this is how much published work in your field is reproducible. So the higher, the bigger bars is better. So you can see that chemistry and physics and engineering are high. So the perception is that a lot of those studies are reproducible. But if you see biology and medicine, that doesn't look, that good. So the crisis seems to be here. And others, as you can see here, that includes psychology, for example, that has been recognized that it has a reproducibility crisis when it comes to reproducing what other researchers have found in that field. Now, they were based, these researchers, obviously it's their own perception. They have not, it's not that they've done studies on this, but basically they asked them, have you failed to reproduce an experiment yourself? And, about 80% in chemistry and biology said, yes, I have failed to reproduce an experiment from another person, but also my own experiments, right? So this is the pink bar there. And, when we haven't really worked in labs, this makes no sense. I mean, you're doing your own experiment, like, why? But there's so many things that, biases and things that can come into play that then you cannot reproduce your own data. So, and as you can see, this actually approaches in some cases more than 60% of people saying, I have not, I have failed at reproducing my own data sometimes. Yep.
Is this for published results?
Not necessarily. Not necessarily. Let me have this one at hand just in case you guys. Not necessarily in this experiment. It's just I have done an experiment, I have a good result, I'm going to reproduce it, and I fail. Not necessarily, but in many cases they have been published. But here it's not required that it's reproduced. And here, as you can see, we have actually talked about all of these issues, right? What factors contribute to irreproducible research? Well, you don't report everything that you did, this selective reporting. Pressure to publish, low statistical power. They didn't do the experiments enough times in the original lab and maybe failed to reproduce. Insufficient oversight, methods or code are unavailable. These days, it's much better if whatever can be made public is made public, including code. Poor experimental design, that raw data is not available from the original lab. Fraud in some cases. I don't think this is, this is a perception, right? I don't think fraud is that common as people perceive it to be, but many of them do think fraud. And also insufficient peer review. We also talked about, papers that, how did this get published? We were discussing that, and it's like, well, maybe there were other interests there, or you're like, oh, this is an important lab, it should get published, and that's a failure of peer review. Okay, and then they asked these same people if they had tried to publish something, an effort of reproduction of someone else's paper. And if they have been able to reproduce it, then it supports the original conclusions. About 24% of these people said, I have been able to publish, and 12% said, I failed to publish. But when it's an unsuccessful reproduction, the numbers go down. So in many cases, I do not know exactly how they conducted this research, this survey, to be honest, but it may seem that you can publish some reproduction attempts, but you still have more successful attempts being published than unsuccessful attempts. And we will see something probably tomorrow's class, if today we may not get there. We'll see about these reproducibility efforts in publication. And finally, if they have actually worried about this problem and have they started procedures for reproducibility in their own labs. And as you can see, everyone is worried, but a third of the people said, well, no, I don't have anything going on really established in my lab to ensure reproducibility of my research. And this is actually complicated because obviously it varies a lot from wet labs to computational labs or hybrid labs. And what exactly is a measurement to ensure reproducibility is hard to say, but this is something that we should be thinking about as scientists, right? Our results, even taking into account everything we've discussed, all the biases we've talked about and how we can fool ourselves, we should still attempt to publish something that we, up until a reasonable, beyond reasonable doubt, we think is correct. Right? And this includes, if you work in labs, try to reproduce your own experiment. So ask a friend or a colleague, can you reproduce this data? And, include them in a team. Something like that could work. Okay, now we are going to start today's activity, and it's going to be a really fun and frustrating activity, I think. We're gonna analyze this paper. This paper was published in 2005. By this researcher, John Ioannidis, who is now in Stanford. Maybe you recognize the name because he was very vocal in the COVID-19 crisis. But this guy published this paper that is one of the most cited papers ever. It's probably the most cited. I can't exactly remember if it's the most cited, but it's definitely up there. And he asked, became really, really famous, and it became, you know, the stepping stone for everyone saying, oh, the reproducibility project, and we need to start, and there's a significant crisis. And as you will see today from this paper, because we're going to analyze it deeply, he concludes that most, not, you know, more than half of research findings in all sciences are false. And other people have you know, disputed this and said this methodology, you know, it has its merits, but it cannot really claim that. We'll see. And we will discuss this, you know, the activity today. So you have this paper now. Is it in the Slack, Mariana? Is it in the Slack, the paper? It's in the class. In the Slack? You have it in the Slack?
Yes, not the instructions. I was waiting for you.
Ah, okay, no, go. Because we're starting here now.
Yes, you have the paper. Okay, so you should be able to access the instructions on Slack. And basically, you have to go to your usual themes. It's very similar to the dynamic that we had for the journal club. You're going to have different sections that you have to focus on. You have 20 minutes to think about it and basically get the main point of what you're reading. It is very important. The paper can get really technical. And it's not the idea that you can get into the technical details in just 20 minutes and with the background that we have given you in this class. So basically just the main point, the big picture, and you're gonna have 3 minutes to present in front of the class. Now, we are gonna be very strict with the time, otherwise it's not gonna be enough, this class. And yes, ask me anything if you have questions, I can explain, but otherwise go to your teams and start working on it. Funny and random anecdote. I cannot pronounce the name of the author. Ioannidis? Okay. I cannot. So if I say your names wrong, just like know that it is a general thing in my brain. 5 more minutes, guys. If you have questions, let me know. Otherwise, think about the main message of your section.
Okay, guys, 3 minutes and we start discussion. If you want us to project something, send it to us in Slack. Or if you just want to write or just talk, just let us know. Time is up. We're going to start discussion. From what I heard, there's a lot to discuss. Good. It's fun.
So
Okay.
yes, let's start. So team number 1, please. And once you are here in the front, you have 3 minutes.
Also, let us know if you want us to project something. Send it in the Slack. Who's team number 1, guys? Who's number 1? Ah, over there. Do you have something that you want us to project or not? No? Okay, okay. Everybody else, pay some attention. Okay, guys, pay attention. Okay, make it.
So,
Okay.
so we could not understand very much in deeper details what he was describing, but basically in the first part of the test, he points that the most part of the papers are only claiming significant results based on p-values, and he's going to establish a way to compare these findings with gold standard, statistical gold standard manner, let's say that way. So he established several values because he says that to find a real truth result, we have to, first of all, have a probability of that. Não vou saber falar. But we have to, there is a probability of that being really true even before we start any experiment. So it's a prior probability of our findings be true. And based on this, let me see what else can I say. People help me.
He said that there are three factors that are really important when we are establishing like a gold standard statistical model to understand really if that relationship is a true relationship. So the first one is the R, that is the number of true relationships with no relationships. And he considers that R over R plus 1 is the true relationships and divided by true relationships plus no relationships. And the alpha values is like a correction to do not include false positives in your analysis. And also the beta is the false negatives correlationships that you can have in your research findings. And by the end, the PPV is the, I forgot the real name, yes, positive predictive values, is the true detected correlations divided by all the true relationships that you could have. And there are some factors, for example, the bias and the number of groups that are researching about that experiment that can influence in this PP value. It's not PP value, but you understood what I said. So, yeah, that's it. And he created a table here that you could understand, for example, if you find a correlationship in your work, and if you really have a relationship. So here is positive with positive, and you have this equation. If you
We can go back to the discussion, but otherwise not everybody gets to discuss it. But yeah, thank you. So to get sure that everybody gets it. So team number 2, please, here.
Okay, so yeah, I guess the best thing to present now would be figure 1, would be the most informative. But so our section focuses on the effect of bias on our yeah. Você quer falar alguma coisa? Porque eu acho que essa parte da disjunção para mim tá.
Eu posso tentar falar essa parte. Figura 2. Você quer botar a tabela?
Pode ser também, mas eu não vejo. Não acho que ela ajuda muito.
Tá
Não.
bom.
Pronto. Good. Yes.
Yeah, so our section focuses on the effect of bias on these positive predictive value of our studies. And so basically, as I guess was in the board already, bias is the effect of just reporting the opposite of the actual result of your study for whatever reason. And when you include the effect of bias, what you see is that, of course, the positive predictive value is going to diminish, right, as you increase the bias. And it's something that is represented in this table, how your PPV is going to depend on the pre-study odds for a variety of predictive powers of your study it's not predictive power, how do you say it? Right, is the variety of beta, the beta parameter, and then for different biases. So as you see here, there's this decreasing bias as you go down these curves, and yeah, as you increase the bias, you see that the actual PPV is going to decrease. And another thing that is discussed in this section is the effect of reproducing the study, so of independent teams reproducing the study. So I guess you can talk about that.
So he talks about the problem of many independent studies studying the same thing, which is kind of strange at the first time because we think that the more the better, the more people studying the same thing. But he says the problem is that those studies usually tend to be interpreted in a disjunctive fashion, in the sense that you have many different studies studying the same thing, and many of them find a false relationship, but people tend to value those that find a true relationship, and that's a problem. And then he just shows that as you increase the number of different studies studying the same thing, the PPV goes down.
Thank you. Team number 3, please.
We can try because dame un segundo. Julietita, ¿cómo estás? Buenos días. Bien, ¿cómo te veo a ti? ¿O más? Depende cómo lo veas. R. Voy. Es 10 sobre el, o sea, es como el total, que son verdades, es 10, es C sobre el total. Sí, acá está.
So yeah, on box 1, they were saying that there's like this study where they were trying to see if any of 100,000 of gene polymorphisms were associated with susceptibility to schizophrenia. And they realized that these 3 tables where they have the normal results, the bias results, and the other one that is just in multiple studies. So basically here you can see the false positives, false negatives, and the opposite. And on the first table you can see that the number of false positive is higher than the number of true positives, and also that happens with the true negatives and the false negatives. And that happens basically in all the tables, now that I see. So you can see that the number of the things that are bad are bigger than the ones that are good. I don't know if you have something to add.
Just to see that in the table 2 we are considering the bias, so you can look for that we are obtaining more like of these expected genes that we have in the first, that we don't have biases.
And in the third table that we see the multiple studies ones, we see that there's less false positives and false negatives. We have more accurate answers. Even with a rank that is particularly strange to me, that is 0 false positives, which was strange. But we see this, and we see a higher number of really negatives compared to false negatives. So In the table 1, more false positives than false negatives. In the table 2 with bias, more false negatives than false positives. And in the table 3, don't have any false negatives, and the false and the positive response is generally more accurate.
It's table 4. It's on the Slack, I think. Okay. So, I don't have the pointer, but basically, this section, he does simulations and obtains a PPV value for based on different combinations of like predictive power that it's this power of the study is 1 minus beta R, which is like some kind of a priori possibility of your findings to be true before actually doing the study, and U, who's like which is like a quantification of the bias. And he says that like he shows in this table obtaining a PPV value that is higher than 0.5 is very, very, very unlikely. And mostly the first two ones are like well-conducted studies that have a PPV value that's 0.85. In the case of the first one, it's a very well-conducted study that has no bias, and it has like a 50% chance of being actually true before you start the study. The second one, he says that it's a meta-analysis. It has very similar parameters, but the meta-analysis, he says, increase the bias. So, but it's still, you get a good PPP value if the studies that you are like actually reviewing in your meta-analysis are actually well conducted and everything. That's like in contrast with the third one, which is the meta-analysis of very badly or small inconclusive studies. There you can see that even a meta-analysis has a very, very low PPV value. And I think that the other part that was interesting is the one that says the adequately powered exploratory epidemiological study and the underpowered exploratory epidemiological study. There you can see that only by decreasing your power, you get like a significant difference in your PPV. And he says that is the same for all these kind of exploratory data-driven epidemiological studies. Most of them are not true, actually.
Thank you.
Perdón que no sé mover tu cámara.
Okay. So he, in this section, he asks, how can we improve the situation? And he is asking, okay, this it's inevitable that most of the research findings are not true, and how can we improve in the future? So he talks about better power evidence, and then he gives a few examples with large scale studies, for example, and low bias meta-analysis. And then he talks examples of large scales. That would be if you're working with, if you're working with data, then you need to have a bigger data set. If you are working with experiments, then you need to have more tests to more experiments to be able to have a more to lower your biases. And but the thing is that not every research is possible that you have large scale. And And another thing that he mentioned is that you should favor questions that they are more general, in a sense that if you have a negative finding, then you can give a feedback to a whole field rather than to a specific question. So you should aim for not too much specific questions, so you can have this more broader view about a field.
Maybe also the registration part, that is important to have standards of registration, because it is hard to collaborate between international groups, but if you register and they all follow the same schemes of registration, it's going to help. That's what I remember.
Well, and the other thing is that he says, like, what is the problem when we rely in one single group result? And the thing is that you cannot dismiss what other research groups are doing in the same field, and what it matters, it's the totality of the result and not like one team result. And that's what basically we saw with the example of yesterday's vaccine. So you cannot give like a huge importance to one single team results. It's just to have a broader view of what people are doing in the same field and reduce biases, I think so.
Team number 6, the final team, please.
Okay. Summarizing, what they say is that instead of only chasing statistical significance, we should also look for our values before starting our research to try to understand what we expect as a result of what we are doing, if we expect it to be positive or negative. And, well, sorry. And also they suggest to when possible redo and try to get the same results as very famous and classical studies and established, and to try to understand if they are really positive, because sometimes we only publish the positives and we cannot know how many times it was done and how much negatives that we got. So the R value could be biased too for the positive results that are published. And you want to talk?
Yeah, basically summarizing that point, the author says in the last paragraph that well-established studies should be redone with large data to see if what was discovered is really true. And he gives a reference based on that also. And the other thing that he also mentions is that the relative, does he say that, like, the relative context in your field should be considered when you try to generalize your results. That's something that's quite common to find the result in a very specific condition and try to generalize that for the field. Want to conclude for us?
Well, so if they've expanded on what essentially is in our part, so I think I'll end on a sad note. Like, well, we begin with the conclusions, and something like the R value is theoretically a very, for me, crazily ambitious quantity to define. I think not even Kant tried to define such a thing, because the probability of truth sounds to me like a chimera, and not really like something that we could systematically apply to a scientific field. And the other thing is the kind of flop-sided way in which the scientific method should be put, in which you should estimate the possibility of truth before you're doing your own research, instead of checking if the results you obtained are statistically significant in the whole scheme of things. It's like I wasn't completely sold on the point of the article, but I didn't read the rigorous part. I think that should be upstairs. That should be up in the paper. So I think that's it.
Thank you. So yes, completely good points. Let's go to the discussion. So just sit down so we can go to that. Thank you. Thank you to all the teams. I know this wasn't the easiest activity. It is a heavy paper, and we were asking you to read just a small section and think about it. Thank you. I think you all did a really good job getting the main point. And hopefully listening to the other teams, you also get an idea, and we can get to the discussion there. I just wanted to mention really quickly here, because I actually skipped in the reading. None of you had to read what the author calls corollaries. For me, it's funny because it's not really derived from what he said. It's just like a random additional comments. But anyway, forgiving him for the choice of words, the comments are relevant and meaningful. Basically, I had my notes. I'm not going to remember by memory. Okay. So basically, just to summarize here, the idea of the importance of the power here is because you have, actually I never put the notes here. So they should be the null hypothesis, and they should be the alternative hypothesis. If, again, like this is a simplification, we are just thinking on one dimension, but it doesn't really matter. The principle is the same, and we talk a little bit about this in the class about p-value. So basically, you have your null distribution associated to your null hypothesis. You have your distribution for the alternative hypothesis. So now you have to think not only on the amount of false positives, this alpha that you want to allow in your test, but you also have to consider that because of the nature of these two things, when you define alpha, you are also defining beta, right? Like if I move this line over here, I get more alpha, and then I let less beta, and the other way around. They are directly related to each other. But also, it is important how actually how different, why my language skills are just really bad, I'm sorry. I cannot say Ioannidis, for instance. Okay, so how different they are from a start is also important, and that comes with your power, right? Like if you don't have a well-defined alternative hypothesis, and sometimes it doesn't have to be about it as your research method. Sometimes it's just a problem, an intrinsic problem of the system, how different they are. And then like how this power is going to be related to this distribution not only depends on, this is the effect that you expect whatever you are measuring to be, that is the difference between these two, but also how fat this distribution is, and that you expect to go with the sample size. If you have and for that, this can be a homework experiment. Just do it thinking about coins or drawing balls from like something, right? Like you don't know, and you're just taking blue and red balls. And if you do that, you're going to see if your sample is bigger, this distribution becomes much more sharper. So how much do you have on this side? The beta becomes smaller, and then your power becomes bigger, right? So it goes with the sample size. It goes with the size of the effect, how different these mu's are from each other. And also, just in general, like how many do you expect to have relationship versus no relationship also is going to affect how much you can do here. That without even taking in account the conflict of interest that you're going to have in your system and all the things that you can decide. Now, before giving the chance you to mention things about this, and just about the last comment, basically, it is true. It is true that we normally have no idea about what is the actual R value. We usually have no idea about what is the true power of your test, but that doesn't mean that you don't have an idea of the range where they live, right? So for instance, and that's why the example of the polymorphisms, and maybe Daniela can actually say much more about this than me, but the example of the polymorphism is really informative, because we already know that there is hundreds of thousands, or at least around 100,000, polymorphisms relevant in the human genome. We know that. And we also know that a disease, general, is not related to all the genes in the genome. It's going to be, even if it is a complex disease, it's going to be driven by a few genes. So we know that from all those 100,000 polymorphisms, only a few, if you're lucky, only a few are going to be related. So that already tells you a lot about the R. We don't know the exact number of R, but we know it's really small, just because of that. And the same thing happens with the power, right? Like if you have a disease, a very dramatic Mendelian disease, then basically you know you have the gene, you have the disease. You don't have the gene, you don't have the disease. So the effect of whatever you're trying to deduce is already huge, and you're going to find it. But if it is something more multifactorial thing, and also like, for instance, like cancer, right? It's not like you have the polymorphisms and you get cancer. No, it increases your chances of having cancer by blah, 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 right? So the size of the effect we know already is not huge. It doesn't matter about your test, it's just about the nature of the system. So we know your power is never gonna be that big, right? So that, I hope that helps a little bit. This is just telling you in general a lot of biological problems that we are trying to solve with data-driven research lie in the region where R is always small and the power is always small, and how do we deal with this? And sorry for my monologue. So do you want to say something?
No, I agree completely on the kinds of studies I work on, which is genomics, large scale. You test all these relationships all the time, and your knowledge already, it's not associated. Like you already know that for the vast majority of them, you're not gonna find a relationship. But people do this, you know, in melanoma, which is what I actually study, we test 1.5 million relationships every time we do an experiment, and so far we've found about 50 that are 50 in the latest meta-analysis with 300,000 people, which is still underpowered for many reasons. And, you know, this is one of the diseases where where a higher number of relationships with the genome have been found. I understand that it's hard to trust everything he says, and we can discuss that now. But yes, we know, as Mariana was saying, that it's a small number that will come out true out of everything that we're testing. At least all the evidence we have so far suggests that is. But that doesn't mean we shouldn't do it. We just should be aware of these things and, you know, how
not overinterpret.
Not overinterpret, and not assuming. I remember the Jelly Beans comic. It's like, you know, one result, one positive result, even if it's a false, that's the one that gets to the media, and that's the one that everyone trusts. The vaccines one. You know, so this is why I guess this title was very provocative. We can discuss if you guys agree with it or not after seeing what they've done, this author. But yes, basically that's the idea. It's vague, but you know, all these biases, misuse of statistics, underpowered studies, small relationships among true relationships contribute to at least many of the published research fundings maybe not being as true as it's claimed in those papers.
So questions or things that you want to add? Anyone?
Do you believe him?
I think I tend to believe him. Then the amount of published works is astounding. So it's probably true that over 50% of them are false, in a sense that the precise claim that they are saying isn't really true, and perhaps a corrected claim would be true, or even it wouldn't be true at all. But I think I tend to agree with him, but when he gets to the math and the corollaries, and I get, yeah, yeah, because I didn't find the theorem he proved from which the corollaries are deriving.
And also complementing that, he also claims all areas, right? I mean,
Yeah, I have a very, very strong issues about that. Like, this is only about p-value research. Like, where is he? Like, he's so much biased towards biomedical research. It is true that a lot of biomedical research is at the end a statistical proof, but there is so many other things in science that don't rely on p-values. So I was very offended, actually, for that part. But I would not believe him the numbers, personally. But I believe the idea and the trend and the issue that he's raising.
Yeah.
I don't know who raised the hand first. Okay.
Just something that I was thinking about because I'm not familiar with the article. I was wondering how was the reaction when it was published, and because for me, when the title say why most published research findings are false, it gives like a very strong claim for people who are very against science, and to use this kind of article to try to argue, okay, I am telling you that we don't believe in science, and then I show you a paper that says that has this title. How can you not believe me? I have this as my to back up my what I'm claiming about not believing science.
you're absolutely true. So the reason why we put this paper here is that it really, really was a big, event when it got published. It's, as I was saying, one of the most cited papers ever in history. And yes, it's been used by some groups to say, well, anyway, scientists are not to be trusted because, look at this guy, he found this. Also, it started this movement, or at least, let's say, gave some legitimacy to these movements of, there is a crisis, a very important crisis, therefore we should do all these efforts, which are not necessarily bad per se, but it was, at least in my point of view, exaggerated in some cases, taken to an extreme of, we have to be super careful, which again is true, but sometimes in some cases it got to the other extreme, right? When maybe, yes, this is a problem, I think we agree, but also many scientists actually responded to this paper saying, he has a right idea, but it's not as dramatic. And some people did follow-up studies and said, actually, if you look at biomedical research, we estimate that only about 20% of research findings are maybe not true. Like they did a different methodology. I can't remember exactly which one, but I've seen the paper. I can share it with you guys. But they did it, other scientists came afterwards and did other different studies in other areas and said, yes, there is a problem, but it's not as big as he is claiming here. So there's been a lot of backlash, and also it's been weaponized by some people to say exactly that. So yeah, I don't know still what to think about this. This is why it's good to hear what you guys think. Yes. Oh, yes.
I had us just like out of curiosity, why are we talking about like a reproducibility crisis now, and like what was the onset of like such a crisis? Because I don't know, when I was like learning, I don't know, physics, for example, and they present you, maybe it's a byproduct of how they present stuff, but there seemed to be no kind of like reproducibility crisis, I don't know, in the 1920s, for example.
Oh, yeah, exactly.
Why now?
Well, I guess someone has, just something. Two things I think relate to that. The first one, as Mariana was saying, when the p-values are invented, which is in the 1920s, but then they become very popular a bit later, people start claiming things based on p-values. Like, that's very popular. And it really became a so-called crisis. I'm still not sure it's a crisis, but yes, there is a bigger problem when, for example, in biomedical sciences, it becomes very easy and cheap to produce massive amounts of data. And then people start claiming any relationship they find among all these tested as something, new. So that's when people actually start realizing, wait, many of those actually have been shown not to be true. And that's when people, at least from my appreciation, start thinking, what is happening? But is on large scale recent p-value dependent biomedical, I would think, data, or big scale data at least.
Yes, this is particularly relevant for data-driven research. And data-driven research in biology is relatively new. Like before, like if you go to the old papers, there is a few that go and rely on data in the most descriptive level. But usually the previous papers are very much experimental genetics, or they are theoretical biology, or things that really don't lie within this area. I think that's part of the problem, and then in less extent, the other part is also how science have moved. Like the number of papers published nowadays is just increasing, not only because we have more scientists actively working, but also the number per capita. Like if you go to the past, like, you know, the very prolific, really good authors will publish a few papers in the whole life, and now it is expected from all of us to publish several per year all the time. So that goes directly to the incentive bias, right? The incentive to prosper in a scientific career, in academic career, goes with the number of papers. So you just want to publish, and then you stop being careful to some extent.
Just a very quick note on that. For example, Frederick Sanger, who won 2 Nobel Prizes for inventing sequencing of DNA and sequencing of proteins, he published like 7 papers? Like it was a ridiculous amount, like super low numbers of papers.
That's what is now expected for you to kind of apply for a faculty position.
You wouldn't survive in Mexico in this environment, you know? I mean, that's the thing. So before, he worked in the 70s. So before, you know, you worked for a very long time on something and then you published a paper. And, you know, that tended to be just because it's a different environment, right? So, yeah, so all of that contributes to most published papers are recent because there has been an explosion, and of those, many of them include large-scale data of some sort. Many of them, at least. Yes,
well, I actually am very glad that you touched in this point, because for me it's one of the things that really comes to the eye when we start talking about reproducibility crisis. That's the ridiculously large amount of papers that are published every year, and that gives us anxiety because we're never going to be able to read all the papers that you want to read, and it's terrible. And I think we are moving from this side of the spectrum where you had very long research in a topic, and then you developed an idea for many years, and then you publish something, and now we have many papers. And I think that somehow what's happening is that we try to sum up over all these and try to get in which direction the vector is pointing, because there are so many papers and you cannot reproduce them all. And the end is like, okay, let's see how many of them got a negative result, how many of them got a positive result, and most of the times is not on the same experiment, not on the same thing, because
so you can do it.
Yes, we're not going to publish the same thing. Reproducing the same thing is very hard to publish this, so it's kind of unsettling, these things.
It's hard when trying to keep up with the literature. I have a Google Scholar alert that sends me papers with keywords, and it sends me every 3 days a very long list of papers. And it's like, I can't, like I read the titles, and it's like, you know, okay, whatever. Like I don't have it, it's a problem, it's a problem. And yeah, but that's the environment we have to do science now. And there's no answer, I think. It's just be aware. Again, this is all be aware.
Well, and reflecting what we can do to better, but no individual is going to solve it. So I want to finish on time today. So go to your questions. It's already in Slack and also here. And we can continue discussion in an informal setting. This is a really important topic, but just to be on time for once.
We can keep tomorrow talking about this, because tomorrow we will see the Cancer Biology Reproducibility Project, which is a very big project that was started about 10 years ago to address these problems. And we're going to discuss this tomorrow.
As soon as you are done with your questions, you are free to go. Thank you.